is our practice, this meeting will be recorded and shared with external audiences. And as you join, I would love to just invite folks to share their title or their affiliation in the chat, just so that we can get an idea of who all is joining us today. And for you all to get warmed up, because we're going to want you to share your questions through the chat as well. And so, as I mentioned, with this being recorded, it will be available on AHRQ's website, which I think is also going to be linked in the chat. So we can start recording. All right. So for those of you who are like, who is this lady? I'm Elizabeth Cope. I'm the Chief Programs and Science Officer at Academy Health. And I also work in partnership with Lucy and others on this to support the Scientific Resource Center. And leading the Grand Rounds session with me today is Dr. Lucy Savitz, Senior Innovation Advisor at UPMC Health Plan Center for High Value Healthcare. And professor at the University of Pittsburgh's Department of Health Policy and Management in the Graduate School of Public Health. And also, fun fact, at least for me, immediate past chair of Academy Health Board of Directors. And so I think we go to the next slide. So we've partnered with AHRQ and the Scientific Resource Center to bring awareness to and help disseminate research findings of the ongoing evidence reports published by the EPC program. Today, the reports that we're highlighting focus on new research in nutrition, which is always, I think, like an exciting area where we end up facing a number of, I think, methodological and analytic challenges. And so it'll be exciting to dig into this with our authors here. And we also will have opportunities for open discussion and Q&A later on, as you can see noted in the agenda. So we do have time here to sort of hear what has come out over some recent systematic reviews. We'll have some reactions from some invited reactors here, and then we will have opportunity for general Q&A and discussion. And so we really hope that you all are thinking about the questions that you have, because we want to make sure we have some lively discussion. So please save those and be able to either enter those into the chat, or I think we also have hand raising features as well that can be used to ask questions. I think on the next slide we have our, oh, this is the CME credits. So at the end of the meeting, we'll share a link in the chat to an evaluation survey that's required for those interested in earning CME credits for this. If we go to the next slide. Oh, did I like breeze through our disclosure?
Yes, but it's okay. It was just in these tables on the last slide.
Oh, okay. Sorry. We did. I'll just say we don't have conflicts of interest to report, so we can move along. So our learning objectives today. Oh, going back to the learning objectives. I'm doing a terrible job, Taylor. Thank you for dancing on this with me. We want to evaluate methodological limitations in the systematic reviews on dietary protein, amino acids, and digestible carbohydrates, including inconsistencies in study design and measurement tools. We're also going to look at current evidence to inform dietary guidance for priority populations, despite gaps and low strength evidence. And then also we want to be thinking about what are our research priorities and emerging frameworks? What are those that are needed to standardize dietary assessment methods and strengthen future evidence? So we have a lot of kind of thorny issues and challenges to navigate together today. So this should be an interesting discussion. And then I would also like to introduce our invited discussants, and we are incredibly thankful that they could join us today. We have Dr. Aaron Carroll, who I don't know at all, despite working at Academy Health. He is our president and CEO and a nationally recognized thought leader, pediatrician, health services researcher, and advocate for the creation and use of evidence to improve health and healthcare for all. He is joined by Dr. Maria Siega-Riz, Dean of the School of Public Health and Health Sciences, and professor in departments of nutrition, biostatistics, and epidemiology at the University of Massachusetts Amherst. Her research focuses on the first 1,000 days of life by understanding the influence of maternal dietary patterns in the etiology of adverse pregnancy outcomes. And then we will also get to, first we'll get to hear from Krista Esslinger, Nutrition Advisor in the Bureau of Healthy Eating Policy and Promotion at Health Canada. She's the lead of the joint US-Canada Dietary Reference Intakes Working Group, and will share details about why these evidence reviews were commissioned and how they'll be used in the future before introducing the authors of the reports being highlighted today. So I'm going to hand it over to Krista.
Great, thank you very much. And thanks for allowing me a few minutes off the top. I'm really happy to be here. As was mentioned, I'm nutrition advisor in the Bureau of Healthy Eating Policy and Promotion at Health Canada, where my day-to-day work is working on translating evidence into policy, including development of Canada's food guide. But today, really, I'm here in my capacity as the chair of the Joint Federal U.S.-Canada Dietary Reference Intakes Working Group. And this is a working group made up of representatives from both of the federal governments, and it really works to support development of the DRI values. And we have been working on preparing to commission a review of all of the values for macronutrients, which are protein, fat, carbohydrate, and fiber. And a key part of this is trying to make sure that all of the needed evidence is available. And so as part of that work, we've commissioned multiple systematic reviews through AHRQ's Evidence-Based Practice Center program. And that's why I'm really thrilled to be able to have a few minutes off the top of this session to provide some context as to why these systematic reviews were commissioned and how at least we intend that the results will be used. So on my next slide, yeah, I've just quickly got some acknowledgements. I mean, I'm here speaking on behalf of a whole collection of people, and really these are some really broad, high-level thank yous. First to my colleagues on the Joint Federal US-Canada DRI Working Group. Then the next three groups are groups that have helped us accomplish an enormous amount of evidence scanning work in just a few short years. So we've got the USDA National Agricultural Library, the USDA Nutrition Evidence Systematic Review Team, and then also the University of Ottawa Heart Institute at the Cardiovascular Research Methods Center. And then finally, of course, the AHRQ EPC program, and particularly the three EPCs that are here today presenting their work. So in terms of what I'd like to do in terms of setting the context, I'd really like to provide you with an overview of the dietary reference intake values, what they are, what is their importance to work in nutrition, describe a little bit about the process by which they're developed, and then talk a bit about the forthcoming review of the DRI values for macronutrients, and then really the role that these systematic reviews will play in supporting the development of those values. So on the next slide, we'll see a little bit about, what are DRIs? They are a set of nutrient intake recommendations that set requirement values, safe upper limits, and values to reduce chronic disease risk for both individuals and populations in the United States and Canada. And so for each nutrient, there is a set of different types of values that are based on different kinds of indicators. And so there's values related to meeting nutritional requirements, and those are based on indicators of adequacy. There's values for preventing excessive intakes, and those are based on indicators of adverse effects. And then values for reducing risk of chronic disease, and those are based on exactly that. And so really the point is that there are all different types of health outcomes that underpin these values, and so there's different types of evidence that are needed to derive them. The other point that I want to make is that there is an overall framework, structure, and consistency to the ways the values are set. One of the key underlying concepts is the recognition that for each nutrient, there's a distribution of requirements in the population. And this really helps to inform the way in which the values can be used and applied. And then the other point is that really in all cases, there should be a demonstration of both a causal relationship and a dose-response relationship between intake of the nutrients and the health outcome of interest. So that's just a little bit about what underlies the values. The next slide will speak to, yes, the importance of the values. And so the figure on the right is a graphic that the Food and Nutrition Board at the National Council of Engineering and Medicine developed several years ago to explain what the DRIs are, why they're important, and you can see that they're a key scientific input to most nutrition policies and initiatives, both within and outside of the government. So things like nutrition labeling, developing dietary guidelines and food guides, making sure there's safe nutrient levels in foods and supplements, creating patient consumer counseling, educational programs, and nutrient intake assessments, and monitoring the nutritional health of the population. So really, these are really fundamental to work in nutritional health and safety. And it's really because of this, that it's important that these values also remain up to date. So on the next slide. Oh, and thanks. Yeah, just keep going. Perfect. Okay, so, oh, sorry. Thank you. So the United States and Canada have jointly supported and funded the development of the DRI values since 1994. So it's a long-standing collaboration between the two countries. And this collaboration is really coordinated through the Joint Federal DRI Working Group, which identifies DRI needs, prioritizes the nutrients for review, and then works to provide the needed funding. And that funding is needed because the two governments actually do not participate in the DRI development process itself. And that's by design. It's because really of the very fact that the DRIs are such a fundamental scientific input to so many activities, it's really important that there's firewalls in place to minimize any real or perceived conflict of interest or bias in the way that the values are developed and what those values actually are. And so both the foundational evidence base to inform the derivation of the values is done independently by having, for example, AHRQ conduct systematic reviews of the evidence, and then the actual DRI values are developed through a process that's managed through the National Academies of Science, Engineering, and Medicine, which is an organization that's independent and at arm's length from government. And so on the next slide, just sort of illustrating, who these three sort of players are that are involved in the process to develop DRIs, and really just trying to show how their roles are distinct from one another. So on the left in the light blue is the Joint Working Group. Again, identifies needs, develops the key questions to frame systematic reviews, and then develops the statement of task and provides the funding. And then all of the other work is done independently. So in the green in the middle, we have AHRQ, which you're familiar with. And then finally, we've got the National Academies of Science, Engineering, and Medicine, which is the body that oversees the development. And the way that the values are actually developed is the National Academies assembles a committee of scientific experts who are then charged to review all the available evidence. They then come to a consensus and then establish the values and produce a report. So really, the roles of these three bodies are really quite distinct. This slide is just talking about how our current project is really the review of macronutrient DRIs, and we've really been working to support this review for a number of years. The macronutrients were prioritized for review based on their relevance to current public health concerns and policy priorities, as well as the availability of significant new and relevant data since the last DRI review. And if you could just advance just once, please. This is really a large and complex project, and that's really because the size and scope of it is just so much bigger than any other DRI nutrient review that has taken place in the last several decades. And it's because of the fact that the macronutrients are all highly interrelated in that your energy has to come from somewhere. So if you have higher of one, you have lower of another by necessity. That each macronutrient is not just one thing, there's many subcomponents to each of them. And then for each of these subcomponents, there's potential DRI values that are related to adequacy, toxicity, and chronic disease risk reduction for all of those and across all of the different age and sex groups. And so there's really a lot of moving parts that need to be considered. And the working group has been working really hard on assembling the evidence that's going to be needed to support and facilitate the review of all of these interrelated nutrient values. So then on the next slide, it's going to talk about a little bit sort of a very simplified schematic of our process. In the last few years, we've really been busy undertaking evidence scanning to identify the biggest data gaps on protein, fat, carbohydrate, and fiber, and then commissioning independent systematic reviews through AHRQ so that they're going to be ready for a committee to work with. And so in the top half of the slide in the purple is showing the macronutrient expert committee, which has not been appointed yet or started any work, but just to show that it's going to be charged with developing all of the values for all the macronutrients altogether. So it's going to look at protein, amino acids, fat and fatty acids, digestible carbohydrates, and fiber all at once. And then the bottom half is the phase that we're in now. That's the evidence review to support the expert committee. And so the DRI working group identified and prioritized questions that need to go forward for systematic review. And as a result, we've commissioned a number of systematic reviews through AHRQ on all of the macronutrient topics. And if you go to the next slide, there's going to be a list of all of these. So when all is said and done, there's going to be 8 systematic review reports that are published in support of the DRI macronutrient project. And so the first 5 on the list, so the protein and amino acids, digestible carbohydrate, and fiber, are all completed, and the final reports are already available. And then the bottom 3 on fat topics are still in progress. And then the 3 on which we're going to be receiving presentations today are highlighted in red. And this brings me now to the part where I'm really excited to introduce to you the individuals who are going to speak to these reports today. And so the first systematic review on the evaluation of dietary protein and amino acid requirements is going to be co-presented by Drs. Maria Butler and Joanne Slavin from the Minnesota EPC. Dr. Butler is with the Center on Aging at the University of Minnesota School of Public Health, and her work focuses on scientific evidence standards, research design, evidence-informed healthcare, and complex interventions. And Dr. Slavin is a professor in the Department of Food Science and Nutrition at University of Minnesota Twin Cities, and her research examines relationships between diet and health, including nutrient needs across the life cycle. The second systematic review on digestible carbohydrate intake and maternal-infant outcomes will be presented by Dr. Diane Stadler, who's professor of medicine and the director of the graduate programs in human nutrition at Oregon Health and Science University. And her clinical work and research have centered on dietary interventions for treatment and prevention of chronic disease. And then Dr. Zhen Wang from the Mayo Clinic EPC will present on the association of digestible carbohydrate intake with cardiovascular disease, type 2 diabetes, obesity, and body composition. And Dr. Wang's research focuses on how current and best medical evidence can be used to help patients, healthcare providers, policymakers, and researchers make informed decisions about care. And so without further ado, I'll pass the floor to Drs. Butler and Slavin to speak to us about dietary protein and amino acid requirements. Thank you.
Thanks so much, Krista. Really appreciate it. And it's an honor to be here. I love to do grand rounds. I'm also a dietitian, so it's really nice to present this and make it available to the public. So appreciate it. Next slide. It takes a village. And as we go through, as Krista just talked about, all these people coming together to support this. These were the co-investigators at the University of Minnesota that helped out. So I'm just having the honor to speak to our work, but we really thank all the funders and the USDA, Health Canada. And I have to say, I teach advanced human nutrition, and I always love the dietary reference intakes just because they are the nutrition Bible. We have to get it right. So when people say, oh, why haven't you changed that? It's a heavy lift. So thank you for everybody being part of this heavy lift. Next slide. So here are our key questions, and they're very big and kind of tough. What are the average daily dietary protein intake requirements of apparently healthy individuals by life stage and sex? And then key question 2, what are the average daily dietary individual indispensable amino acid intake requirements of apparently healthy individuals, again, by life stage and sex? Next slide. So our methods. And I would love to have Dr. Butler here because she's the expert and really was the PI. So I was a nutrition person, and but I'm lucky enough to get to present it. So where did we go at our database that we looked between January 2000 and May 2023, looking at those sites that are listed there, and anything we could bring in. So as we would find new information, bringing that in with hand searching whenever possible to find information. It was really hard as you find this, that there aren't going to be, I always want to mention that protein is not a nutrient of concern. The average person in Canada and the US gets enough. So there's not a ton of new intervention studies where we're just looking at protein requirements. So, we wanted to only have human studies, prospective cohort, any nested case control studies, and getting across the life cycle from infants all the way through the elderly, and anything on how we could get data, what were we looking at, and nitrogen balance, indicator amino acid oxidation, just because the standards for this in human studies really aren't, do we have a new method? Do we have a new method to do this? And so nitrogen balance, you'll see we sometimes that's the best information we had, even though it seems really old school. It is some of the stuff we were looking at. Risk of bias is a new concept for me, but it was really tough to come up with studies that did not pass this. So we did lose some studies based on that, if they weren't randomized, things like that, and then trying to grade it and making sure that we only had information included that was up to the standards. Next slide. So here's just, it's a narrative qualitative synthesis because it was not possible to do a meta-analysis. We didn't have enough in any group to actually pull it together that way. And so the first is population, and then anything we could find on individual amino acid groups. These population groups are the ones that are in the DRI. So this is the way it's divided up anyway. And if we could get an estimated average requirement is how we scale up to get an RDA. Whenever we could get that, we would provide that, but sometimes that was not there. So appreciate that in pulling this together. A lot of the studies we saw did not have the information. Next slide. Results, keep going. So here's the literature flow. And you saw that huge list of people that helped in this. It was a very heavy lift to do the initial, is there anything new since 2000 in protein requirements, amino acid requirements, and then sort through this information. So all of this is in the report too. So we'll keep going here. So this was, what did we find? And you think, oh, in 23 years, that's it. How can this be? But for question protein, 9 of 21 studies were assessed as low to moderate risk of bias. And of these, 7 reported a protein requirement estimate. So it seems like with all that work, why couldn't we have more? And remember too, with all these different groups. So looking at, the types of studies we could include and what kind of information we got for infants, children, pregnant, adults, that is the summary of the information that was available. And if you go over to the outcomes reported, I mentioned it would be great if these studies all had the same outcome, and some of the newer outcomes, newer ways of measuring protein metabolism were in these studies, but generally you'll see we had nitrogen balance or some other methods that were hard to collapse down and compare. Next slide. Okay, so now we get to amino acid. 31 of 38 studies were assessed as low to moderate risk of bias that were included. And as you might expect, a lot of the studies when we're looking in this area are going to be exercise studies because people are much more interested in amino acids in an exercise study that would be actually published as opposed to just we're going to figure out amino acids. And if you go back, how did we learn about basic nutrition? Amino acids studies are basically done in animal models, and those are not part of our review here. So these are the number of studies where we had information on amino acid requirements in healthy humans. Next slide. Some of the key points, and this is just a number of examples and the summaries, the outcome that was reported. We can just kind of high spot this, but infants, there was 1 randomized control. And then they actually calculated phenylalanine requirement from that. In children, aromatic amino acid requirement was possible from the 1 RCT that was out. 2 pregnant studies that we came up with, phenylalanine and aromatic amino acid requirements in that. In adults, there were 3. And it's really hard. And as I mentioned, typically in a human study in the US, we're not deficient in amino acids. We're not deficient in protein. So it's hard to come up with estimates of requirement for that. And then in adults, a little bit on phenylalanine. Next slide. So if we go and say, well, what I think a lot of people would be very disappointed is like, hey, but this is the important thing to update what's in the DRIs. What's the new information that adds to the body of evidence? Do we have data that we have new information that would tell us that we need to change the dietary reference intakes for protein? So what's the good thing about what we have is we have a well-conducted systematic review of a wide range of population groups, including older people, pregnant people, and any methodology we did include it all. Obviously, most of it was in the nitrogen balance, just because that's what was there. And that we did get rid of studies that were, they were excluded because of risk of bias. Some of the methods, and if you get into this, a lot of the new methods that are out there are considered better. And some of that research, a lot of it, animal nutrition studies, people on animal nutrition products showing that there's higher quality proteins, different ways of measuring it. A lot of these methods are pretty expensive and you don't see them a lot. So generally, nitrogen balance is something that is kind of the standard, even though it doesn't seem very high tech, but we didn't have enough of these other newer studies, newer ways of analyzing to give us any way of using that and comparing it to nitrogen balance and saying that the data we have from nitrogen balance over the years is not useful. And we mentioned just most groups, only 1 or 2 high quality studies. So there's very limited evidence, and it really doesn't give us any amount of data that we need to change the current DRIs for protein and indispensable amino acid requirements based on what we found in our systematic review. Next slide. And then future reference needs. I do a lot on protein quality. I teach a lot on protein quality, and what you've already heard about is that protein is just another energy source. So when you're trying to do these studies, if you're not controlling for energy, if you're on a low calorie diet and protein is one of your calorie sources, you're going to use it for calories rather than building body tissue. So it's really hard to look at protein needs. Protein quality, another huge issue as more people are getting into plant proteins. I mentioned that protein has never been a nutrient of concern in the US and the Canadian population. Most people get enough of it. But as people are going to lower quality protein with a deficient in 1 or more amino acids, that becomes real important, and we don't have a ton of research on that. Gender differences, very few studies get into that. And is there a standardized method? It still kind of goes back to nitrogen balance as the one study. For the last 100+ years, that's what we had, just because we have the ability to measure that and estimate protein in, protein out from that. And then just more, typical diet patterns as people change diet patterns too. In the usual diet patterns, we have so much protein that we're not going to see deficiencies. So we really can't get at any new information. As people do switch to these different proteins, more plant proteins, lower quality, it's really important for us to focus on that and get more research in that. Next slide. I appreciate some initial reactions from our panel. Thank you.
So I don't know who wants to go first. Aaron?
Sure. I'm just sort of struck by the fact that, as you say, that there are, it's not a nutrient of concern, which makes it very hard to do this kind of work. So part of me is, interested in, like, if we were going to design a study or really think about how would we go about getting this, how would we do that?
Yeah, that's really difficult in a human population. Just because if you want to see, a lot of times if you want to see needs, you put people on deficient diets to see if you see differences. So on a calorie, if I have enough calories, I'm healthy. If I generated my protein, and I go back to what the original DRIs are, the 10 to 35% of your calories should come from protein. So if you don't control calories, you're never going to find anything in these studies. So, and I would probably recommend, if we're going to fund research in healthy people, a lot of interest in the elderly, and really not much studies at all, hard population to study. Same thing in pregnancy and babies. So I just think that we don't want to, this is why we've done animal studies forever. If we want to stress the system and kind of figure out where the stress points are, we do it in an animal model just because of those concerns with putting a human at risk. So I think humans are quite, they're quite available to whether their protein is 10% of their calories or 35%, they can do pretty well on that. And it would be hard to come up with a biomarker to show, to kind of fine tune that any more than where we're at. And we know what the essential amino acids are, and generally too, if they're eating complete proteins, we're not going to see a amino acid deficiency.
So I'll give you some initial reactions. First of all, thank you for the excellent work. Having done systematic reviews, there's a lot of work that goes into them. And the key questions really guide your work. And you had very focused key questions. And so as a nutrition researcher in the field, I'm not surprised that you haven't found that many studies done since, the 2000s. And that's because our research focus in the field has been different. So I would honestly say for the last 2 to 3 decades, we've been more concerned on understanding dietary patterns and the effects of dietary patterns on chronic disease and overall health outcomes. And that's because, as you said, we haven't seen deficiencies in protein that we were concerned about. We sort of have come, we've evolved so much more, and that is not an area of concern. So it doesn't surprise me that we don't really see any newer information in that particular field. And the other thing that I think is really important, and this is going to be true for each one of these systematic reviews, is the way we're looking at scientific evidence right now to inform nutrition policies, such as the DRIs and the dietary guidelines, is much more rigorous. And so therefore, conducting these systematic reviews now are very different than the way we did it in the past in order to come up with the DRIs or the dietary guidelines. And I think that's really important. And I thank Krista for the way she framed us at the beginning of this session to really understand the importance of how we need to have good scientific evidence, and for everyone to understand that the field has appreciated that, and our tools and our methodology for what we now consider in the evidence has changed, and it's gotten stronger, and that's a good thing. And last but not least, I think, and I hope we can talk about it a little bit later, just as you mentioned, Joanne, what does the future work need to look like? And if there is future work, and every single one of these reports is going to identify this future work, I would really hope that both the Canadians and the United States equivalent of NIH or USDA takes that into consideration and really write specific proposals to drive future research to be able to identify the current research gaps that we have in order to come up with strong scientific evidence to inform our dietary policies in the United States and Canada. I'll stop there.
I appreciate that comment, because I always tell my students, if you get enough energy, you're going to get enough protein. So dietary patterns really bring protein together. So if people are eating enough calories, you can complement your plant-based proteins and do really well. And I think that's where consumers are going. So to me, I completely agree with you that as a classically trained nutritionist, it's like, well, there must be more out there. Where are these studies hiding? But why would we're not putting people on deficient diets to look at a biomarker. So we're probably not going to see those studies. So appreciate the comments.
So I would follow up, because I think this ties back into sort of my initial question, because it does not surprise me at all that, you look through the literature and we see what we have, and it's a lot of bias studies. It's a lot of methodologies that might not give us the answers we want, but we keep seeing so many groups speak with such certainty about what we should be eating and in amounts. And I'm just sort of at a point where I'm thinking well, what study would it take to definitively answer that question, so that we don't have to keep sort of doing this over and over again? And it may be animal models, and it may be something else, but it really would be good to really get concrete guidance on what question needs to be answered, what study would actually get us there, and then really direct us in that direction.
I completely agree. And I always tell my students that humans are omnivores, so we're really flexible, but number one, get protein every day. Get water, number one, and then get protein. So I really appreciate protein getting to lead off here, because to me, the fat and, it's basically the fill after that. So we want to make sure that everybody in the US and Canada has access to adequate protein, and along that with calories, and then we can fill the rest of the calories later. So And that, as we go, people eat food, so it's got to be in dietary patterns too, rather than just grams of protein and blah, blah, blah. So I think we're moving in the right direction. I'm pretty positive.
All right, I think we're moving to our next presentation with Diane.
Great. Thank you. Let's go ahead and go to the next slide. And again, we have no relevant disclosures. Next slide. And we too had a village. I like that analogy. It takes a village to create this type of work. And the names on this slide probably don't represent everybody that had their hands in this project. And I am here as a representative of this group and completely appreciative of the amount of work that went into taking on and completing this project. Next slide. So I want to provide a little bit of background before we dive into the actual review that was done here, and that focused really on digestible carbohydrate during pregnancy and infancy and maternal and infant outcomes. And we know that optimal nutrition during pregnancy is essential to support placental fetal growth, and during infancy to promote appropriate growth and cognitive development. Nutrition during these life stages has long-term health impacts extending beyond the immediate period of pregnancy and infancy into adolescence and adulthood. As Anna Marie mentioned, as she was being introduced, the first 1,000 days, the concepts of the developmental origins of health and disease have their origins prior to pregnancy, during pregnancy, and early infancy for those long-term health outcomes. Foods containing carbohydrates are really important sources of energy during these life stages, but more important is that the foods that contain these different types of carbohydrates are excellent sources of micronutrients, and we need to keep that in mind as well. And that's one of the reasons why it's so important to look not only at the amount of carbohydrate that's consumed, but also the quality of the foods that provide those carbohydrates. With respect to the DRIs for total carbohydrate, the recommended dietary allowances during pregnancy currently are 175 grams of carbohydrate, total carbohydrate per day. Although this current recommendation does not take into account the requirements for placental function, so that is being reviewed and considered at this point. The RDA for childhood individuals older than 1 year of age is 130 grams of carbohydrate per day, and that is the minimum amount of carbohydrate that actually extends beyond childhood into adulthood for the minimum amount of total carbohydrate. Adequate intakes during infancy have been established at 60 grams per day between birth and 6 months of age for healthy infants, and 95 grams per day for infants between 6 and 12 months of age. And again, these are healthy individuals without any type of complicating conditions or metabolic conditions that might impact the utilization of carbohydrate as an energy source. The DRIs for total carbohydrate include both digestible and non-digestible carbohydrate, and our review really focused on digestible carbohydrate. And these DRIs were published in 2005 and were established without using any systematic review methodology. And there are no specific recommendations for the component of total carbohydrate that is comprised of digestible carbohydrate. Next slide. So I wanted just to, in a very simplistic way, describe how total dietary carbohydrate is categorized into digestible and non-digestible carbohydrate. There are many ways that carbohydrate can be subcategorized, and this is one way that total carbohydrate can be categorized into digestible carbohydrate, which includes the monosaccharides, the simple sugars, including glucose, fructose, galactose, and other monosaccharides, the disaccharides, maltose, sucrose, lactose, and other monosaccharides, polysaccharides, which are commonly referred to as starch, long chains of monosaccharides that are present in a number of different food types, and then sugar alcohols, which tend not to be digested in a similar way, but can provide calories and can be taken into consideration. And digestible carbohydrates are distinguished from non-digestible carbohydrates in that they're readily digested, absorbed, and enter into our metabolic systems, whereas non-digestible carbohydrates, including plant-derived fibers and animal-derived non-digestible oligosaccharides, which are starting to receive a lot of attention, are a type of carbohydrate that were excluded from our particular systematic review. Next slide. So the purpose and the key questions that we explored, the purpose was to identify and systematically summarize the evidence published since 2000 on the relationships linking digestible carbohydrate intake that was consumed during pregnancy to newborn growth parameters, items such as weight, height, head circumference, frequently measured growth assessments, and size at birth, which might be considered large for gestational age, appropriate or small for gestational age, and gestational weight gain of the pregnant individual. And then the second category of questions we looked at was, when digestible carbohydrate is consumed during infancy, what are the relationships between infant growth, infant size, and body composition from birth to 24 months of age? Next slide. And so the methods that we used, very similar to what you've heard in the prior presentation and what you'll hear in the upcoming presentation, is that we followed the guidelines of AHRQ that are well established and create the basis for many of these systematic reviews. We used a priori defined study eligibility criteria that were established by our partners and our technical expert panel. We searched a number of databases that looked at the evidence that was published between January of 2000 and April of 2025. We independently and dual reviewed all of our studies against these study inclusion and exclusion criteria. We included both non-randomized or randomized controlled trials and prospective comparative trials. We synthesized the studies qualitatively. In this case, for the same reasons mentioned before, we didn't have the number of studies that were appropriate to be able to do quantitative meta-analysis. We graded based on strength of evidence. We looked at risk of bias, and then the protocol was published in Prospero. Next slide. Looking at the eligibility criteria, these are just some of the highlights that were used. We explored the literature that was available for generally healthy individuals during pregnancy and during infancy and young childhood. Specifically, exclusion criteria included not looking at children that were born prematurely or individuals that developed or had been diagnosed with gestational diabetes as one particular exclusion criteria. We had to be able to identify digestible carbohydrate. We had to be able to dissect that out from total carbohydrate, or it had to be reported as digestible carbohydrate. And what you'll see in the next few slides is that the utilization of the term digestible carbohydrate is not used frequently in the scientific literature. Primarily, total carbohydrate is published, fiber might be published, Added sugars might be published, but we had to be able to identify digestible carbohydrate. Our comparators, yep, there you go. Our comparators were different types, or excuse me, different amounts of digestible carbohydrate between different groups. The outcomes were standard measures of growth and gestational weight gain. Timing had to be during pregnancy or from birth up until 24 months of age. Our studies had to be done in free living individuals and outpatient types of settings in countries with high or very high human development index scores. The study designs, as I mentioned, randomized control trials, non-randomized studies, prospective cohorts, nested cohort studies. The sample size had to be adequate, which in many situations, having a sample size of 30 in a randomized controlled trial, either of infants or of individuals during pregnancy, can be a high bar to reach, especially if you have to have more than 10 individuals per study arm. And then the publications had to be written in English and published during or after 2000. Next slide. So our strength of evidence was based on methodological limitations of the studies, which were identified through our risk of bias, the imprecision of the studies that might have been present or identifiable in the study descriptions, the indirectness of the evidence. And so we found that many of the studies that were published or even the studies that we included in our systematic review weren't done to answer the questions that we were asking in our key questions. So information might be available, but it wasn't directly related to the questions that we were trying to answer. The consistency or inconsistency of the results, the heterogeneity of the data that was available, and then the likelihood of reporting or having publication bias were the components of the strength of evidence that we evaluated. Next slide. This slide illustrates our literature flow diagram. At first look, we had more than 3,000 studies that were identified as meeting some of the criteria. On first review, the number of studies, almost the majority were excluded during the abstract review. We went deeper into more than 300 studies for our full text evaluation. And after excluding based on full text reviews, we had 8 studies in 10 different publications that we included in this particular systematic review. Looking at the 3 different key questions that we had for the maternal intake and newborn outcomes question, we had 6 of those 8 studies were relevant for key question 1. Additional 6 were relevant for key question 2 that was looking at maternal intake and maternal outcomes. And for the 3rd key question that looked at infant intake and infant outcomes, there was only 1 of the 8 studies that was relevant for that question. Next slide. So the 30,000 foot look at our study findings was that there really weren't any studies that reported differences on newborn size when there were different amounts of digestible carbohydrate consumed for our infant population, and the strength of evidence was low. When we were considering newborn adiposity, both size, body composition of infants between birth and 24 months of age, there was no data available to evaluate differences in the amounts of digestible carbohydrate intake. And there was no difference in infant adiposity from those studies, and the strength of evidence was insufficient. Thinking about infant size at birth, adequate, appropriate, large, or small for gestational age, there were no differences in those outcomes with respect to differences in digestible carbohydrate intake. And again, the strength of evidence was insufficient. And then for maternal gestational weight gain, there were the majority of studies that were evaluated, there were no differences in gestational weight gain or minor differences in gestational weight gain if there were differences in the amounts of digestible carbohydrate consumed. And again, the strength of evidence was low. So with the next few slides, we're going to dive a little bit deeper into looking at what that evidence suggests. Next slide, please. So this is for key question 1, maternal digestible carbohydrate intake and infant birth outcomes, those growth and size markers. And you can see when we looked at weight as the outcome variable, the strength of evidence was low or not available to be able to answer. You can see that oftentimes the number of studies that were available to specifically address a certain question was 1, and it was 1 non-randomized study. And you can see that in most cases there were similar rates of weight gain or rates of low birth weight or percentages of infants that were born small or large for gestational age. With respect to length, there was 1 randomized trial, 1 non-randomized trial. There were no significant differences in length as an outcome. Same thing for head circumference. There was only 1 study that addressed body composition or fat distribution among the infant birth outcome population. And then for BMI and other composite measures of body composition, there were no studies available linking maternal digestible carbohydrate intake to infant outcomes. So very limited evidence available to draw these relationships. Next slide. For the second key question that was looking at maternal intake and maternal outcomes, where the maternal outcome was gestational weight gain or a comparison of weight gain with respect to what the recommended weight gain was for certain individuals. You can see that there were 6 different studies that we were able to analyze to answer this question. And again, the strength of evidence of the studies was low or not applicable given the few numbers of studies. And when you look at the summary findings, there really weren't any differences in gestational weight gain with respect to maternal digestible carbohydrate intake. And if there were differences, the differences were relatively small, and it was difficult to draw conclusions. When we compared weight gain to the recommended amount of weight gain, there were no differences in either achieving the recommended weight gain or in the percentage of the sample that had inadequate weight gain throughout pregnancy. Next slide. And then our third key question was looking at infant intake between birth and 24 months of age and infant outcomes, looking at growth, various growth outcomes. Again, weight, length, head circumference, and various body composition measurements. And you can see here that the strength of evidence for this key question was insufficient across the board for the different outcome parameters. There was 1 randomized controlled trial that was deemed to be appropriate or relevant to this question. And you can see that there were no differences in weight at 9 months of age. In one particular study, there were similar birth weight z-scores. There was no difference in birth length. There was no difference in head circumference at 9 months of age, and there was no difference in body composition as assessed by BMI at 9 months of age. Next slide. So what are the relevant findings from this systematic review? Well, some things that we found as we were reviewing the literature, and this really contributes to our recommendations for future studies, is that with the current literature that's available for us to review, the trends towards, and Anna Marie brought this up, the trends towards reporting dietary consumption patterns really limited our ability to dissect out specific nutrients, and in particular, digestible carbohydrate, based on the way information was being reported. So dietary consumption patterns, instead of isolated nutrients is really influencing the way we could answer the key questions that were posed for our systematic review. The other thing that's really interesting is there's a strong tendency to use principal component analysis as the statistical analysis method, or one of them, that is being used to categorize and analyze dietary data. And again, that limits our ability to look specifically at certain nutrients and their impact on outcome variables. The limited evidence for this review reflects the fact that most of the published studies did not use the term or did not define digestible carbohydrate in their publication, and they didn't allow us to have the data that we needed: total carbohydrate, total fiber, and other subclassifications of digestible carbohydrate to come up with a value that would represent total dietary carbohydrate intake. Next slide. So some of the specific limitations, if you think about the limitations of the types of studies that are reported, we were looking at the pregnant population and the very young infant and early childhood population. And these are two different populations that are, essentially really hard to study. Pregnancy in humans lasting over a 9-month period of time is a transition period over that 9-month period of time. So oftentimes we would see studies that were reported that assessed dietary intake at one point in time during pregnancy, the second trimester, the first trimester, but often not frequently over the course of pregnancy. And similar challenges in being able to assess dietary intake of young infants and during early infancy, early child, 1 to 2 years of age, also posed significant limitations in the ability to draw conclusions. The focus of our some of the limitations, not being able to quantify digestible carbohydrate, and similarly not being able for future studies to be able to identify the source of that carbohydrate and the quality of that carbohydrate. So I think those are going to be questions and requests that we'll have in the future to be able to tease out what the relationships are between digestible carbohydrate and these outcome variables. I mentioned not using the term digestible carbohydrate, and also I think we would say from a published literature standpoint that just the variability, the heterogeneity of the studies and the published data that is available during pregnancy, and the methods that are used to assess dietary intake, to assess human milk consumption among the infant population, and the composition of human milk and the volume of human milk consumed are limitations in being able to really tease out those relationships. Next slide, please. So some of the research needs that we found as we were going through this literature, as of course, targeted rigorous research needs to be done on these topics. More very strenuous research needs to be done both in the pregnant population and during infancy. One thing that we identified was we feel that this information is available. When you're doing dietary studies and you're looking at dietary analysis, you are creating databases that have, in most cases, all of the different macronutrients and micronutrients and subcategorization of those macronutrients. And so we feel that in most cases, the information that we need to answer these questions is out there. It just hasn't been reported. And that's confirmed by the fact that most of the studies we looked at, the questions that were being answered were not directly related to the questions that we had in this systematic review. So being able to have funding available to reanalyze data sets that might be able to answer these questions would be a strong first step in future research. And then additionally, new studies rigorously designed that are really looking at dietary intake over the course of pregnancy teasing out the nuances that occur in the first trimester, the second trimester, and the third trimester, being able to identify how those differences might impact gestational weight gain or infant growth, birth outcomes, or infant growth early in those first 2 years is really important. And then extending those studies into the postpartum period and being able to link up maternal ability to successfully feed their infants through human milk, and the composition of that human milk is critically important. And those are severely limited at this point in time. Next slide. Great. Thank you very much. We really enjoyed being a part of this series of systematic reviews, and especially being able to identify those future research needs, which we know can be accomplished. So thank you.
So happy to offer some quick reactions. But first, Diane, you didn't leave me much, because you actually did a really nice job of not only, identifying, what you did, but also the implications of the findings and where those limitations are. And I just want to stress a couple of things that you mentioned, which one is the fact that these are very special populations. And in order to really be able to study them requires great sophistication in our methodology. I mean, infants with the intake of human milk is very, very hard and tedious, and it takes a lot to even, if you can do those studies. And then during pregnancy, having done the pregnancy cohort, several of myself, the question is, how can I assess dietary intake during that time period to be representative? And the reality is the majority of us use food frequency questionnaires. And then when we use food frequency questionnaires, we know that when we think about getting absolute intakes of carbohydrates, protein, and fats, that's limited. We know that, those are not perfect numbers whatsoever. And while we have moved to getting 24-hour recalls once a month or a couple during, a particular trimester, that even has limitations in and of itself. So very, very complex, methodology for answering a question of such high importance.
Right.
And so while I agree with you, there's data out there that might be able to address digestible carbohydrates in some manner, because that's not a term that we've used in the past. I'm not sure that we would have the capacity to feel confident in determining absolute amounts of carbohydrates, fats, and proteins to answer these particular questions of interest. And the only thing I'll add here is that while you were trying to address digestible carbohydrates, and I know that, there were a couple of other systematic reviews that were done for the dietary guidelines related to dietary patterns and some of these outcomes that you reviewed. And even with that systematic review, there was limited evidence for understanding a relationship between dietary patterns and excessive gestational weight gain. And there was limited, evidence for looking at dietary patterns associated with lower risk of developing gestational diabetes. And we found no evidence for hypertension. So, some of the things is very similar. You can sort of see, the evidence sort of following one another. But once again, our methodology is limiting us here.
I agree. I also think, whenever I'm working with investigators and they're planning their projects and we try and create that perfect study, dietary analysis, carrying out through frequency questionnaires, 24-hour recalls, it takes a lot of time and labor, and they're expensive. And people will elect not to include dietary analysis because of the added cost associated with that. And we have to be able to figure out a way, a method that allows us to efficiently and in a valid way collect information that is not limiting the budgets of these, comprehensive studies because we can't collect dietary information that would align nicely with it. So that's another complicating factor. Yes, thank
And
you.
the fact that we don't have biomarkers for each one of these either.
And again, just as before, excellent methodology. I have no quibbles about, how everything is done. It's great. But I'm struck by, as a pediatrician, like if we had any group that we could probably study really, really well, it might be young infants. I mean, breastfeeding obviously is going to be difficult, but otherwise, the vast majority of young infants are just having formula. Where we know exactly how much of, digestible carbohydrates there are. And if we could just measure how much formula you had in an accurate way, we would capture 100% of the nutritional intake up until they start eating solid food. And even then, it's not that hard because so many people are just feeding out of a jar where, again, it's 100% measurable, or that the number of foods being consumed is relatively small. So I would hope, no ding on you, I would hope that we would have, of any group of humans, the best data on infants who are 0 to 6, which makes you almost more frustrated that we're still struggling so hard. But again, I'm just sort of, I'm coming back to the idea of like, we got to get better, I think, about, saying these are the studies that we need, and to continue to gather studies which aren't helpful to us is really a waste of money and time, and that we really need to zone in on exactly what would fill the gaps.
Mm-hmm. And I think you're right. There are studies, there are formula-based studies, and many of those studies were done prior to 2000.
Yeah.
Right? Many of those studies were done in the 80s, the 90s. And, there are a number of studies that are still being done with formula-fed infants and tweaking little components to the formulas, the different types of fatty acids, different protein content or types of protein, different sources of carbohydrate. And it's just really interesting to see that many of those studies are still being done to some extent in that very young population. And if we had any studies that had high sample sizes, those were the studies that were available to us. Yeah.
And I just would add one more thing. I think if anything, that the vast majority of clinicians and parents are like, well, actually not parents, but clinicians certainly much more concerned about overconsumption than underconsumption. And so, less failure to thrive issues than there are like, are we priming obesity in this population, which is a completely different set of concerns, and trying to figure out these requirements in light of all that has got to be enormously difficult.
Absolutely. Good point.
All right, let's move ahead. This has been a great discussion so far, and I look forward to the Q&A that comes to follow. So I will just put in another plug and reminder. I know a few folks have put questions in the chat and want to encourage people to still keep doing that. We'll come back to that. But let me turn us over to our third presentation.
So I'm happy to present our findings on these two systematic reviews. Next slide. First, disclosures. And the studies are supported by AHRQ. I have no conflict interest to disclose. Next slide. First, I want to acknowledge and thank our wonderful AHRQ task order officers, David and Holly, and then our amazing partners from HHS and the USDA, Julie and Juliet. Without your help, and these studies will be much difficult. And I also want to acknowledge and thank our technical expert panels. Next slide. First, a little bit background. As we know, type 2 diabetes and cardiovascular disease are severe public health concerns with high impact on human life and health expenditures. Many studies have reported that the increasing prevalence of type 2 diabetes and cardiovascular disease worldwide. In the United States, CVD remains the leading cause of death, while 11.6% of US population have diabetes. Of these, 90% to 95% of them have type 2 diabetes. Personal and cultural dietary habits have been identified as potential risk factors, particularly carbohydrate intakes. Healthcare authorities and international health organizations have published guidelines for optimal consumed energy percentage or quantity of carbohydrate intake. Despite some inconsistencies in the methodologies and contests, the recommendations are generally similar and recommend carbohydrate intake to be between 45% and 65% of total energy consumption. The only exception is the WHO. WHO recommends up to 75% of the energy to be from carbohydrates. Next slide. So the DRI for carbohydrates were published in 2005 and were essentially determined based on the brain's estimated requirement of glucose in different age groups. For children older than 1 year old and adults of all age groups and sexes, the R.D.A for carbohydrates is set as 130 grams per day. Next slide. Since the publication of 2005 DRI, and there have been significant growth in the body evidence. So we conducted two related systematic reviews to address the following two questions. The association between dietary digestible carbohydrate intake and the incidence of cardiovascular disease. And then the second one, the association between dietary digestible carbohydrate intake and the incidence of type 2 diabetes and the effect on growth, size, and the body composition. Next slide. So these two systematic reviews for the established methodology of systematic reviews as outlined in the AHRQ method guide for effectiveness and comparative effectiveness reviews. Essentially, we defined the study eligibility criteria. We conduct a comprehensive literature search, conduct abstract screening and full text screening. We extract data and conduct meta-analysis whenever possible. We assess the risk of bias using the Robbins-I tool, and then grade the strength of evidence. Next slide. Brief introduction of the eligibility criteria. The first one for cardiovascular disease. Basically, the eligible studies have to meet all of the following criteria. General population over 2 years old, report total dietary digestible carbohydrate intake and isolate the effect of association of digestible carbohydrate intake from the effects of other macronutrients, and have to compare the carbohydrate intake level to another a different total dietary digestible carbohydrate intake level or levels. Report the outcome of interest, cardiovascular disease or intermediate outcomes, and then follow the participants for at least 4 weeks. We include randomized control trials, non-randomized control trials, prospective cohort studies, and the next case control studies. The studies need to be published in English as a peer-reviewed full-text publication, and we only include the studies published after 2000. The last literature search was completed in July 2024. Next slide. For the second review, the eligibility criteria are quite similar to the first one, except that we only we are interested in the outcome for type 2 diabetes, gestational diabetes, growth, size, body composition, and some surrogate markers. And then the studies need to follow the participants for at least 12 weeks. Otherwise, the criteria are very similar to the previous one. Next one. So we graded the strength of evidence using the following 5 domains: methodological orientation of the studies, in other words, risk bias, imprecision, indirectness, inconsistency, and increased likelihood of reporting the population bias. Next slide. So for the first review on cardiovascular disease, we started with over 9,600 citations, and we ended up with 21 original studies from 29 articles. These 21 studies include over 1.3, about 1.3 million participants. All these 21 original studies are prospective core studies, and we found no relevant clinical trials. Most of the studies were conducted in the United States, Europe, Asia, and one study conducted in Australia, and one in multiple continents. The median follow-up for participants were 10.7 years, ranging from 1 year to 32 years. Next slide. The median age was 51.7 years, ranging from 25 years to 58 years, and we found no studies evaluated children under 18 years old. About 60% of the participants were women, and the median digestible carbohydrate intake was 51%, ranging from 33 to 79%, or 252.7 grams per day. The median protein intake was 16.2%, fiber intake 21.9 grams per day, and the fat intake 33.4%. Next slide. These two figures show the non-linear dose response association between carbohydrate intake and cardiovascular disease. The right figure analyzes the carbohydrate intake as a percentage of total energy intake. It showed a gradual decrease in the risk of CVD up to 50% of total energy intake, followed by an increase at higher carbohydrate intake levels. Compared with a reference level of 50% of total energy intake, carbohydrate intake exceeding 65% was associated with a significant increased risk of CVD. Similarly, analyzing carbohydrate intake in grams per day, the left figure revealed a steady decline in CVD risk up to 250 grams per day, with an increased risk of at higher carbohydrate intake levels. Compared with 300 grams per day, exceeding 300 grams per day was associated with a significant increased risk of CVD. Next slide. These two figures show that the nonlinear dose-response relationship CHD. The right figure showed a relative flat risk of incident CHD up to 45% of total energy intake. However, the risk rose sharply after that, and compared with the carbohydrate intake reference level of 50%, exceeding 50% of total energy intake with half of carbohydrate intake was associated with a significant increase in CHD risk. Similarly, the left figure revealed a flat risk up to 250 grams per day, and followed by a sharp rise in the risk at a higher carbohydrate intake levels. Next slide. So to summarize, there was a nonlinear association between digestible carbohydrate intake and cardiovascular outcomes. And we rated these outcomes were rated as a low stress evidence. So compared with reference intake of 300 grams per day or 50% of total energy intake, CVD risk was significantly increased when intake exceeded 300 grams per day or 65%. The lowest risk was observed when carbohydrate intake was 250 grams per day or 50%. CHD risk also increased when carbohydrate intake was 250 grams per day or 45%. We found a U-shaped association of the carbohydrate intake and the CVD mortality. The lowest risk was observed where carbohydrate intake was 250 to 300 grams per day. And stroke was not significantly associated with the carbohydrate intake. There was also a less defined dose-response relationship. We conducted a multiple subgroup analysis, and the subgroup analysis based on sex and geographic locations found very similar results. Next slide. So this figure shows the flow chart for the second review on type 2 diabetes. We started with 13,000 citations, and we included 17 original studies from 23 articles. A total of 500 participants were included. 15 of the 17 studies were prospective observational studies and the 2 randomized clinical trials. However, these 2 randomized trials were presented analysis like observational studies because the comparisons were from these 2 trials are not whatever we wanted to evaluate. So majority of the studies were conducted in United States, Europe, Asia, and the 2 studies were conducted in Australia. Similar to the previous review, the median follow-up was 10 years. Next slide. Median age 49 years. Also, we found no studies about the children under 18. The majority of the participants were women. 76% of the participants were women, and the median carbohydrate intake was 49%, and 294.9 grams per day. These numbers are very similar to the previous review. Next slide. These 2 figures show the nonlinear dose response analysis for type 2 diabetes, and we found a L-shaped association. So analyzing carbohydrate intake as a percentage of total energy intake showed a graduate reduction in the risk of incident type 2 diabetes up to 45% of total energy intake. The risk then remained flat between 45 and 55. And after that, risk increased. Similarly, analyzing carbohydrate intake in grams per day, the left figure, revealed a gradually reduced risk up to 270 grams per day, followed by a plateau between 270 to 350 grams per day, and then increased after that. Next slide. 7 studies with about 20,000 participants reported body weight. And however, for this outcome, we were not able to conduct a meta-analysis. And so 2 studies reported reduced weight associated with the increasing carbohydrate intake. 1 study reported significant increase in weight, and the 4th study reported non-significant difference. And so for this outcome, we rated as insufficient. Next slide. So to summarize, there was a nonlinear association between digestible carbohydrate intake and type 2 diabetes with low strength evidence. Type 2 diabetes risk gradually decreased from the lowest intake until 45% or 270 grams per day. The risk remained relatively flat between 45% and 55%, or 270 grams per day to 350 grams per day, and then increased after that. Subgroup analysis based on sex and geographic locations found similar findings. Next slide. So to conclude, we found a non-linear relationship between the intake of digestible carbohydrates and cardiovascular disease outcomes and incident type 2 diabetes. A specific range of carbohydrate intake was associated with a lower risk. The current literature suffers from a lot of limitations, and I think Diane gave a really good summary. this limitation also applied to our findings. So most studies were inadequate or deemed inadequate to adequately adjust confounding. Also, studies were unable to clearly isolate the effect of macronutrients from each other. And then, like all other systematic reviews and meta-analysis, our findings are subject to ecological bias. So that concludes my presentation.
Thank you very much. I'm just checking to see. I think at this time, we're actually going to move into some general Q&A. And so I know some folks have been putting things in the, have been putting some things in the chat. Now looking through there, but I think so. At this point, we will we'll start with questions for our authors, and then in a moment pivot over to talking with our discussants for a little more discussion there. Just kind of, I think, kick things off, and then I'm going to go see if I can mine some additional questions through the chat. But if I sort of going back to that first report that we were talking about. If there's a way to think about, because I think this is a theme that has been recurring through the different reports that we've been hearing about, which is sort of the challenge that we have in the overall sort of consistency and quality of the evidence, and then thinking about how do we connect this, right? How do practitioners navigate the guidance and think about how they should proceed? And so if we think about populations where protein needs are critical, if we think about infants and those who are pregnant or lactating, how should practitioners maybe approach some of the guidance given the current uncertainty and lack of standardized methods? Like, how do we sort of navigate that sea?
Well, I'll go first here. Being a classically trained nutritionist, we do know nutrients and we know a lot. And so we know protein is the most important macronutrient. We have to get it. And during growth and development, it's particularly important. And over time, we've had access in this country and Canada to really good protein sources. So it hasn't been limited. And that's one of the reasons it hasn't been studied as much as we might like. But I believe that we don't need to move away from nutrients. Right now, we have all this push on nutrients and getting away from processed foods and food, it's like all this push. And as was said, if you look at infant formula, there is really high quality protein that is in that infant formula, and it's very standardized. So we've created some important things, but consumers have lost kind of confidence in the food supply. So a lot of people are leaving and saying, we're going to do this ourselves. And because they've never taken a basic nutrition class, they don't understand protein quantity and protein quality. So that to me is what, and then we say, well, plant protein's good, animal protein's bad. It's like, no, we don't want people to go there too. So I think on the practitioner side, we have to make sure for everybody, protein is the most important nutrient, and it's an expensive nutrient, and we want to make sure everybody gets it. And so therefore, even though some of the chat too, why would we expect there to be studies out there where we decrease protein a little bit and see a big change in biomarkers or some kind of disease risk? So I just think we know a ton, but we still have to say, hey guys, when you are out selecting a product for your kids during pregnancy, during lactation, even if you're like, I'm pregnant, I don't want to eat, make sure we get protein in 3 meals a day and the highest quality protein we can. So I don't think the messages on the practitioner side have changed too much. I don't know what people would expect. We don't really want to see, because, when we heard about wasting, it used to be, if we go back when we said, hey, get rid of fat in kids, we know that kids need high fat diets. So then parents say, hey, I want to do right by my kids, and they take it out of their diet. And then we do have failure to thrive in some of the most well-off people in the world. So I just think to kind of take a step back and encourage people to appreciate the food supply we have, and protein, make sure 3 meals a day for your kids and your family that you can provide it for them. And if you can't, we need government programs to help you.
Thank you. Oh,
I just want to also say, you know, when we were first designing our review on carbohydrates, we spent a lot of time talking about protein and fat and you know why the question as it was asked for the US and Canadian populations with respect to carbohydrate, we spent a lot of time talking about protein and what are the because protein is a nutrient of concern in many other countries, and carbohydrate is not that nutrient of concern. It is the staple in many other countries. And one of the limitations of the reviews that we were doing is we're really trying to identify information that is relevant to the US and Canadian population, whereas many you know, policymakers, governing bodies, and other countries look to the United States for or the US and Canada for our recommendations to try and implement similar types of guidelines in countries where protein may be limited, and carbohydrate intake may be the real predominant source of calories, of energy. And I remember those conversations were really interesting as we tried to tease out why is it that we're looking at digestible carbohydrate in the US and Canadian populations at this point in time, given that the diet is so there's so much access to those types of foods. So
great. Thank you so much. I was kind of scrolling through, and it looks like people have been really wonderful about responding to questions in the chat. So, but I did want to just see if Anjali Jain is able to come off mute, perhaps. I know there was a comment in the chat about being struck about how we're using disease-based models of research for a more wellness-based interest. And I don't know if you want to be able to speak to that a little bit more so we can hear from our panelists.
Sure. It's Anjali, by the way. Thanks, Elizabeth. Yeah, I think that part of the reason we want to know, you know, sort of the optimal amount of protein, for example, and I think this is a question that's out there in the public space quite a lot, right? And so we're trying to ask about wellness, really, rather than deficiencies or what's not enough as much, except for in the special populations, which is where the studies are being done more. So I think it would just be interesting, like Erin, I think your comment about like, how would we study this? And I think getting the question a little bit more pinpointed, and perhaps the outcomes separated between sort of optimal wellness, meaning energy to perform, for example, wakefulness, attention, those kinds of things. I just feel like we're trending in a direction of wanting to know those kinds of outcomes, and yet we're, you know, our sort of way of doing research is very disease-based. And so I think that some of that gap is really calls for some out-of-the-box thinking of how we might actually answer some of those questions a little bit better. And, you know, I think that there's a huge wellness community that's probably monitoring all their intakes pretty well. And, you know, maybe Apple has that data and we don't. But I do think that kind of data might be useful to explore and to try to get some sense of, like, not only how do you not be deficient, but how do you actually improve some of these outcomes, which, you know, frankly, our country is suffering from more than many others because of, you know, maybe processed foods or stress or all, you know, various different aspects, right? So that's all.
I'd love to jump in here because I think we forget the RDAs are recommended dietary allowances. So, they're very high. So the RDAs, when we look at the DRIs, the RDAs are part of that. So they're really high. And I guess, this is my consumer side going, there's this huge interest in protein and people are taking ridiculous of protein and taking protein supplements. And there's no information at all that is necessary. And protein is also not a nutrient of concern. So to try to get that, I agree, it's a wellness question rather than a disease prevention, that if you just take in too much protein, it just gets deconstruction. You got to get rid of the urea and use it as like a carbohydrate or a fat. And you can get very fat on excess protein, which seems pretty obvious, but somehow consumers don't get that. So this current just complete ridiculousness of more protein, put protein everywhere where it never was before, is relevant. And I think that's what this review helps, is that we don't have any data that there's a protein deficiency and that we need more additional protein. But it's really important to think of the groups that we've been trying to focus in on here: pregnancy, infants, during growth and development. Anytime there's growth, protein needs go up, and we want to make sure the highest quality protein is available then. So it's a little bit hard, just because I agree with you. The dietary guidelines are set up for disease risk, exposure to diets, where the DRIs are really make sure we don't have deficiencies and try to get the best numbers we can for these very diverse groups with really different food supplies out there. And we have to believe that, a lot of people don't breastfeed, and that infant formula is actually a pretty good product. And we could do studies on that, but those studies might be funded by people that make infant formula, but that might be some of the best research we have too.
I would also, just to add to that, is there is a lot of data that infant formula companies do have that is not publicly available, and it's not available in published forms. So how do we better partner with private industry to make sure that we have access to as much information as possible to be able to, just saturate our data sets and our evidence?
I mean, just one added note. I think most of the people on this call probably understand what high quality protein is, but I don't think the public does very well at all. And so it would be interesting to get a little bit more messaging out there about that.
And even our labeling, we have tons of details on carbohydrates and fats on our nutrition facts label. We have one line for protein. And that's just grams of protein, and there's nothing to do with quality. So I agree with you that even if they wanted the information, it's kind of hard to get it.
I'm going to come in with just one more question for our colleagues working on digestible carbohydrate intake here, and then move along to Lucy and discussion with our reactors. But just in terms of the research that was presented there, we had two reviews that were discussed. But in looking at those data, we ran into a number of issues with particular gaps in the studies and some methodological challenges, particularly related to confounding adjustment and those kinds of things. And so in both of those cases, if you had to prioritize sort of the one either main gap that really needs to be addressed as a top priority, or one sort of methodological challenge that needs to be confronted, what would you say that is?
Well, from a digestible carbohydrate standpoint, I think when we report data in publications, or when we have solicitations for specific types of studies, I think we need to identify that we want more than just the primary total carbohydrate reported. So total carbohydrate, fiber, we want more. I think to answer these questions, we need more discrete data that is presented. And as that has come up in the chat, we're not talking at all about quality. And, we can, divide carbohydrate into simple and complex carbohydrates. And that's interesting, but really what we need to understand are what are the sources of the carbohydrate. I mean, Are the carbohydrates coming from whole grains, fruits, and vegetables that are providing other nutrients alongside carbohydrate as an energy source? Or is it coming from simple sugars that are not providing any of those micronutrients as well? So while I want to understand the discreteness of digestible carbohydrate, I also want to know the complexity of not just how much, but what else is packaged with that food, and the interconnectedness of all of those nutrients in promoting wellness or preventing disease.
Yeah, it's hard to ask a lot of questions if you can't access granular data. Exactly, yeah.
From my perspective, so I believe that larger population-based prospective course studies with long follow-up would be really necessary, and a rigorous long-term prospective study in a controlled feeding environment would address a lot of limitations we face in this study. But obviously, it's unlikely to do that. And so the intake of life plan span may influence the development of chronic disease. Studies following individuals throughout their lifespan would be ideal. However, a more reasonable or feasible approach would be maybe follow up for at least 5 to 10 years. And such study should look at other aspects of the nutrition matrix, not just the carbohydrate per se, and monitor weight stability and weight change in addition to other lifestyle factors. Certainly, dietary assessment methods or validated tools should be used with better collection data and adjustment of these co-founders. So I think these are important for us to address.
I like that. I think we definitely want to have more like 30-year-long studies that collect all the data, right?
Yeah.
Yeah.
Ideally.
Easy problem.
She is being sarcastic, I think.
Yeah. Well, thank you very much. I'm going to turn things over to Lucy and our reactors for some additional discussion.
Great, thank you. And this has been an incredibly rich discussion. And what that means is we've run close to time. So we have a little over 10 minutes to come back to our discussants, Anna Marie and Aaron, as we think and really sort of reflect on policy, practice, and health systems implications based on what we've heard from these reports. And so I'll go through the 3 reports and ask each of you to comment. If you think there's some other point that you think is important to make based on this rich discussion, please feel free to go ahead. But in terms of the protein and amino acid requirements report, I'm just wondering, Anna Marie, from a systems level perspective, how could the current lack of conclusive evidence affect broader health outcomes related to child growth, maternal health, and nutrition equity? And what role should policymakers play in addressing these gaps?
So, I mean, I think what we saw is the fact that there isn't sufficient evidence at this particular point in time to change what the recommendations are. And the recommendations were based on classic studies that, did do things in a particular way. Now, I'm going to say this, and it's really true, is that the data that they used for those reports in the years of 2000, 2005 did not go through the rigorous systematic reviews that we're trying to do right now, right? And a lot of the recommendations for like infants and children were extrapolated from adults. So we have to keep that in mind. So there is no doubt that we need new questions and new studies to address them. But I think from a policy perspective, it's important to understand that we're not seeing more deficiencies. We know what protein deficiencies look like. And you can detect protein deficiencies. If you go to a pediatrician, like Aaron's going to be able to detect a child who is protein deficient. And so I think that's where us sort of setting those guardrails in terms of practice of understanding, okay, where are the deficits? What are the signs of these deficiencies associated with these nutrients? Have they been identified? Are they increasing or not? And then the excess. and the excesses usually relate to some kind of a disease.
Mhm.
So that's why we monitor, relationships with CVD, with type 2 diabetes, and with protein, we haven't even talked about, the stress put on the kidney. So, I mean, I think the reality is, from a practice point of view, as a practitioner, what you're looking for is that overall health and wellness of your particular patient. Are they coming in? Are they worried about their diet? Have they gained too much weight? Are there metabolic markers within the normal range? So we're using tools that will help us identify whether or not there is an issue that needs to be addressed with a particular patient or population that you're particularly addressing. And I don't think that has changed yet. And I think that's where we need to keep the messaging consistent, because while we don't have new information to address these DRIs, we have information on overall dietary patterns that assesses not only the pattern, but to some degree the quality too of those particular foods. And we do have recommendations out there that are based on scientific evidence that is really strong and moderate. And so that doesn't necessarily change what we're telling our patients and what we're informing our policies on right now.
Very helpful. Thank you. Aaron, your thoughts?
I have so many, and it's hard to even know where to start. But I guess if I was just going to try to, squeeze it down, especially since of this discussion, I'm trying to think about what are we trying to answer here? And the truth of the matter is that if we're talking about like protein and amino acids, it's like we just want to make sure that we're not getting too little, which is going to be pretty rare, or you're not going to be getting way too much, which could be harmful. And we can talk about it in terms of whether it's wellness or whether it's a disease-based focus. But I think we're trying to tell people, like, you don't want to go too low, you don't want to go too high. And the truth of the matter is that the range is enormous in between there about sort of what's acceptable, because unlike, I think, the way that a lot of people are trying to do that, it's not as if you can dial in an optimal amount and say, if I could just hit this number, I'll live forever. That's not going to happen. And to really get down to, like, I really want a number and I want it to be statistically, I'm not sure we're ever going to have it. I'm not sure it's even possible. I'm not sure we'd want to have it. And I think, again, sometimes I think as just stepping way back, we lose sight of what we're really trying to do, which is like, is there an identifiable number which is too low and you got to be dangerous? Yes. And is there just way too much? There's probably. But after that, I wonder if sometimes we should just say, other than that, just, eat lots of foods and you're going to be fine.
Yeah, Aaron, I would like to say, to some degree, I'm in total agreement with you, right? And I wanted to ask Dr. Shang in particular about the evidence related to CVD and the digestible carbohydrates, because it represented 1.2 million people. That's probably the largest number of individuals for which we have data on. And yet the evidence was rated as low.
Yeah.
So, is the evidence rated low because of all the confounding associated with those particular studies, because there's multiple cohorts here? And is there a way that we as a scientific community can go back to the authors of those cohorts and say, can you all answer the question like this? With 1.2 million people and them having collected dietary data, can we not get to the answer and feel more confident with the answer?
I'll take it even a step further, because I had further questions about that specific one, because there was the typical, which we see in nutrition all the time, U-shaped curve. But it seemed like with carbohydrates, there was almost a willingness to accept, okay, well, obviously too low is bad, too high is bad. But if you put that same graph up there with salt, and it absolutely exists, people discount the low end of the curve. They say, no, that's an artifact. It's confounding. We should get it down to zero. With alcohol, you sometimes see that. And we say, no, that can't be real. That's got to be confounding. it's the same U-shaped curve we see almost with everything in nutrition, because it probably is some confounding. That, you know, people on the very low end of the spectrum have other things going on, but I'm struck that if it's something we think is potentially positive, we accept it. But if it's something that we, for a variety of moral or other reasons, think it's bad, we say, no, 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 that's confounding, ignore the bottom end.
Very interesting. Zhen, I don't know if you want to comment based on you being sort of called out here.
Yes.
Oh, I'm not in any way suggesting.
No, I don't mean
that
called out in a
it's-
negative way. I mean
Oh, yeah.
referred to.
Yeah, I mean, it's just,
One
it's
choice of
reality.
words. Yeah.
No, I could no that, and so yeah, and so the issue with the literature we've identified, and they suffer multiple limitations, and I don't know. I'm not sure if a reanalysis of the existing would address a lot of issues. One of the problem with that is you know the validity of the tool, dietary assessment tool or methods used in these. And in these studies, sometimes it's really hard to tell whether they are good or not. And so certainly, and I'm not sure if they the original studies they collected all the necessary data, and to enable us to do better analysis, to better adjust the confounders. But certainly for the future studies, and we certainly can do a lot of things better, and a lot better. And but for the existing studies, I'm not so sure about that. But yes, for the so that's the reason we used data that presented us, and we did the meta-analysis from the U-shaped curve, and we certainly incorporated this uncertainty here and rate as a low stress evidence. And so that's really going down to how we are going to interpret the findings, and we are going to how to use these findings in the practice, right? So we certainly need to incorporate individual risk, and we certainly I think most people agree that the moderate for this nutrition intake would be ideal for general population, and for those patients with additional risk, and the individualized approach would be necessary.
This is in no way a judgment of your methodology, of your findings, but I'm struck. But if we're talking about how are we going to take this to the next level of policy, that if we look at your curves, we would say, okay, look, between this is where it starts to seem to level out, and this is where it is. So this inside is the optimal level of carbohydrates. That's what we should be consuming. But if I take that same chart and it's with salt, and I say, let's look at mortality with salt, and the sweet spot for the lowest all-cause mortality is like between 3 and 6, if I remember correctly, like grams per day or something like that. But every guideline in the world for policy shoots the number far lower than 3. The WHO says like 1.5. I think the CDC is like 2.1. So and that's because people are like, yeah, I know that the sweet spot on the graph is here, but we almost like don't even believe that. We still think it should be here because it's salt. But for carbohydrate, we're interpreting it as like, no, the sweet spot is the sweet spot. And I'm struck by like how, you know, why do we say that the sweet spot is here if it's carbohydrate, but if we take the same chart in salt, we say, no, we got to shoot far lower.
I don't think he can answer that question.
No, I'm talking about, so this is a broad discussant point about policy and how we take it.
Absolutely. But I do think one of the things that, and I think this is brought up in the questions, and all of us would definitely agree, there's so much intra-individual variation in how we all consume our nutrients and how they react in our bodies. And until the day that we really have what's called precision nutrition, really, well characterized, because right now we don't, and I don't believe using, any kind of like a Google search or entering data into Google will help us, because there'll always be that self-reporting bias. that, we'll just have to take some uncertainty related to dietary intake and the policy that comes along with it, because we don't have those tools of exact precision. And quite frankly, there's very few tools, even in medicine, that are exactly precise. And I just think we have to acknowledge that and accept it.
Lucy, I believe you are muted. But it was very animated.
Yeah, yeah, I'm high energy. This is a great discussion. There's a lot of work that still needs to be done, but we appreciate all the work of the authors and their teams in preparing the reports and presenting them to our discussants. Thank you. And I'm going to turn it over to Elizabeth to close us out.
Yeah, I know it's a bummer to come in on a lively discussion and just be like, we're done with this. I think it's fun when we can have a lively debate and also be thinking about, right, where are the ways that we can sort of challenge ourselves in the science to improve and to be better, and where are there places where we sort of have to recognize that, like, we all are dealing with sort of imperfections and challenges in science, and this is part of our journey together. So I appreciated the good discussion there. But we are coming to the end of our time on this grand rounds, and I just want to thank everyone for joining us and participating in today's conversation. I want to recognize our report authors that joined us and had 3 fantastic reports to talk through, as well as our discussants, Anna Marie, and Aaron, as well as our partners at AHRQ and the SRC. I believe, if it hasn't gone in there already, oh, there it is. We've got, Taylor has added the CME survey link in the chat for anyone interested in seeking continuing medical education credits. Just as a reminder, we will post the meeting's recording as soon as possible and announce the next EPC Grand Rounds theme shortly thereafter. Thank you all again for wonderful participation and wonderful discussion today. Fantastic reports. Thank you, and I hope you all have a good rest of your day and a wonderful week.
Thank you.